back. This is Dr. Mobeen. We are continuing with our questions around immunology for the topics which we have covered so far. So today is the time for the lecture or the question number 11. So let's very quickly look at the question. So the question is very simple. A patient with the ulcerative colitis responds well to a bacteria which has been introduced in his gut, and that bacteria is secreting a recombinant interleukin or a cytokine or a chemical substance, and the patient is responding to it. And the question is, what interleukin may be producing this effect? And the options are interleukin 2, interleukin 12, interleukin 4, and D, interleukin 10. So the question is, out of all of those, which interleukin is responsible for this particular action? So really, if you think about it, in this question what they're trying to ask you is, you know that the ulcerative colitis or Crohn's disease or asthma or organ transplant or leprosy, these are the examples where the body's immune system has taken a route which is not the correct route for responding to some sort of pathogen or foreign substance. So there is an anomaly in there, there is a problem in there. So in this particular case as well, the person has ulcerative colitis, so that means his immune system is reacting a little bit more, and then we have provided a bacteria there in his gut which offers some chemical substance which is helping. And the question is, what help and how? So out of these options, which one is it? So let's see. We're going to go back to the bigger picture and see what are the chemical substances released by various cells and what is their effect, and then we'll come back to the answers. So let's start for how to attempt this question. So the very first thing to notice is that, so let's start from a nice macrophage. Of course, these are nice cells. So monocyte, macrophage, a cell which is present here, and you know that the macrophages have gotten receptors. So we've gotten the Toll-like receptors, we've gotten the complement receptors, we've gotten mannose receptors, then we've gotten scavenger receptors, and others. So there are some receptors which macrophage uses to identify the pathogen in the extracellular spaces and then phagocytosing them. So you know about that. Once the macrophage has phagocytosed some bacteria or a pathogen, then we also know that it fuses that with the lysosomal vacuole, and finally the bacteria is killed and sliced and diced and broken up into pieces, and those pieces are then externalized on the surface of the macrophage as on MHC2. And remember that whenever this externalization or antigen presentation comes, there is a co-stimulatory molecule named B7 protein. So MHC2 and B7 are working together to present the antigen to the cells which are going to work with it. So primarily the helper cells, T helper cells. So that is one thing to remember. The other thing to remember is that when the macrophage phagocytoses something, so remember this thing, this is a very important concept here. You need to probably make a box around that and put some stars. When a macrophage does phagocytosing, it releases interleukin 12. On the other hand, if the macrophage is doing phagocytosis and then has become active because it has been activated by which chemical substance? Gamma interferon, right? Gamma interferon coming from the helper cells. I'll draw them in a second. But if the macrophage has become activated, then the activated macrophage releases interleukin 12 and also releases tumor necrosis factor alpha. In addition to that, you also are aware that the macrophage releases IL-1, IL-6, IL-8, of course, and then you know it also releases granulocyte, monocyte, colony stimulating factor, and many other factors. These are just some factors which are important for us to understand for today's lecture, or to answer this particular question. Now let's say if we go in the sequence of events, pathogen has been engulfed, phagocytosed. It has been fused with the lysosome, and so there is the phagolysosome, and IL-12 has been releasing, and the antigen has been presented on MHC2. Now this IL-12 goes and acts on which cell? Remember, T helper 0 cells. T helper 0 cells. It would go and act on the T helper 0 cell, and the IL-12 causes T helper 0 cell to become what? T helper 1 cell, remember? So this is the T helper 1 cell, and the function of the T helper 1 cell, what is the function? Number 1, it releases IL-2, interleukin 2, and number 2, it releases gamma interferon gamma. So remember, this is the IL-12 interferon gamma axis, which we have done in the other previous lectures. The important thing is that we have got an IL-2 over here. This IL-2 actually acts back onto this T helper 1 cell as well. Now IL-2 then also works on which cell? Remember, the cytotoxic T cells. So this is a cytotoxic T cell. What does that mean? That means this is CD8 cell. CD8. This is CD4. This is CD4, right? That is CD8. And CD8 cell, the cytotoxic cell, is ultimately going to work with the cells which have gotten the pathogen in them and then try to kill those cells. Correct? So that is the basic function here. So from pathogen IL-12 moved the T helper 0 to T helper 1. T helper 1 then caused the cytotoxic cells to be activated. On the other hand, it also released the interferon gamma, which caused the macrophage to become activated as well. Activated macrophage. And I hope you understand that by activated macrophage what we mean. What that means is that the number 1, the receptor expression on the macrophage has reduced. Number 2, the reactive oxygen species have increased. Number 3, reactive nitrogen species have increased, and then overall activity of the macrophage has improved. The receptor expression has reduced. So that is an activated macrophage which has become activated with the interferon gamma. On the other hand, now if we come back here, you would also know, and I have covered that in our previous lectures, that T helper 1 can become T helper 2. T helper 1 can become, I make these cells as they're wearing t-shirts. So this is T helper 2. It can become T helper 1. T helper 0, or the naive T cell or null T cell, can become T helper 2 cell. Again, this is a CD4 cell. Can become a T helper 2 cell in the presence of interleukin 4. Right? And we know, we have discussed that in the past, that interleukin 4, we do not know which cells, which ninja cells are hidden and releasing the interleukin 4 to convert the T helper 0 to T helper 2. But in turn, once we've gotten the T helper 0 becoming a T helper 2, we know that T helper 2 releases IL-4 and IL-5, which then works on the B cells. And then, so let's say this is a B cell. I usually make the B cell like this, right? A little chemical factor, and releasing the antibodies, right? So this is the B cell. So that is the pathway here. This is humoral pathway. I have repeated this so many times, I hope you remember it. This is the cytotoxic or cellular pathway. Cellular pathway. And remember that cellular pathway not only activates the T cells, cytotoxic T cells, it also activates the macrophages. So the macrophage, which is here, is also activated by the cellular pathway. So this is also cellular pathway, right? Now the question is, which interleukin over there is applicable here? Before we go there, I'm going to put one more cell here, and that is our nice fellow the natural killer cell. Remember, I make the natural killer cell like this. So this is that pervert which I've made many times, which is working. So this pervert, remember, it palpates and it checks out the surfaces of every kind of cell. But the important thing is that the NK cells are part of the innate immunity, while the cytotoxic T cells are part of the immunity for the cellular immunity or acquired immunity. So this all is acquired immunity. This is innate immunity. This is also innate immunity. I normally make the natural killer cell somewhere over here, but for our discussion today, I'm going to leave it there because we have to work on it. Now, most of these interleukins here are actually pro-inflammatory interleukins. That means what? That means that these interleukins are working to cause inflammation. The question here is asking that the patient actually recovered, or patient not recovered felt better. So that means that inflammation, that extra immune response has become improved. That means they are asking that are you aware of anti-inflammatory proteins? So the anti-inflammatory proteins or regulatory proteins or suppressors of immune systems, there are two primary, there are many, but there are two primary substances which are really important, and these two are tumor growth factor beta, released by macrophages and monocytes and many other cells, and interleukin 10. Interleukin 10 is released by many cells as well, but the most important cell which releases the interleukin 10 is the T helper 2 cell. This cell releases interleukin 10. And I had covered it in the past that this interleukin 10 release is actually to tip the balance of immune system towards the humoral as compared to cellular. So let's see very quickly what is the function of interleukin 10. Interleukin 10 is anti-inflammatory. Tumor growth factor beta is anti-inflammatory. Remember them, just these two. Just remember these two, IL-10 and TGF-Beta. These two are suppressors. So wherever there is inflammation and then you want to suppress or control the inflammation, these two proteins are really, really important. That is what they're trying to figure out, that are you aware of which protein. So of course, if you think about it, interleukin 2, 12, 4 and 10, of course the 10 is the one which is there. There is no other interleukin, all other are actually pro-inflammatory. So let's just very quickly see what that means and what do these things do. Interleukin 10, one of the functions, it acts on T helper 1 cell and reduces the activity of T helper 1. So that is one function. That simply means that the cellular reaction pathway would be suppressed. Interleukin 10 also acts on the natural killer cells, and what it does is it reduces the interferon gamma production from the natural killer cells. Natural killer cells produce interferon gamma. So that is a second. Now here in the case of the helper 1 cells, it also does this at the interferon gamma. This is the negative effect here as well. Interferon gamma secreted by the T helper cell is also reduced. And then very, very important thing, interleukin 10, when it acts here, when it acts on the macrophage, it reduces the GM-CSF, granulocyte monocyte colony stimulating factor, so it reduces further production of these monocytes and macrophages. It reduces tumor necrosis factor alpha. Do not forget these things, write them down somewhere. It reduces the secretion of tumor necrosis factor alpha from the macrophage. That is the second thing which it does. Then this is very important, it reduces the expression of B7 and MHC2. It reduces the expression of the B7 and MHC2. What does that mean? That means that this macrophage will not be able to present the antigens very successfully, or even if it does present, it would not be able to co-stimulate the cell, the helper cells. So that is also a very important effect which is happening here. So reduced GM-CSF, reduced tumor necrosis factor alpha, reduced presentation of pathogens, and then it also, very important, it also reduces negative effect on IL-12. So interleukin 12 is also reduced, which would mean that further upregulation of the immune system to start defending is also deregulated or dysregulated. So at the end of the day, what did interleukin 10 do? It reduced the natural killer cells action, it reduced the activity of the T helper 1 cell, it reduced the activity of the macrophage. Not only reduced the activity of the macrophage, it actually reduced the amplifying effect of macrophage on the remaining immune system. And so what has happened now is that it has successfully suppressed the inflammatory proteins and the cycle. The other one factor which does these kind of things is tumor growth factor beta. Tumor growth factor beta, I'm going to draw it with the green marker. The green marker somehow does not look very clear on these videos, but anyways, I want to separate that from the things here. TGF-Beta released from macrophages, it is released from monocyte, it is released by many cells. Most important thing to remember about TGF-Beta is very simple. That is, TGF-Beta's function is to reduce the proliferation, differentiation and activity of the cells. That is the primary function. Proliferation, differentiation and activation of the cell. So the very first function TGF-Beta does, it works on the macrophage itself and it reduces the activity of the macrophage. So that is the first function, reduces activation of the macrophage. That is one function. Second function is it acts on the natural killer cells, and what does it do? Reduces the activity of the natural killer cells or differentiation of the natural killer cells. It also works on the T helper 1 cells, and what does it do over there? Same thing, it reduces negative effect on the T1 cell, it reduces the activity of the T1 helper cells, and then finally it acts on the B cells and reduces the secretions of the B cells. So that means humoral response is also reduced, right? So you're seeing that the innate response is reduced, you are seeing that the response here for the helper cell and the cytotoxic response is reduced, you are seeing that the pathogen response is reduced, or the humoral response is reduced, and then you're seeing that the innate response here by the macrophages is reduced. So that is the effect of tumor growth factor beta. What is the function of the TGF-Beta? TGF-Beta is going to reduce the activity, differentiation and proliferation of various cells. Most importantly, B cells, natural killer cells, T helper cells and macrophages, monocytes themselves. So these are the two primary proteins which would do this. So if we went back to the question and try to figure out what was happening. So again, what is most important here is interleukin 2. That interleukin does not have, interleukin 2 has nothing to do with the suppression. Rather, interleukin 2 is actually the one which activates the cytotoxic T cells, and it activates the helper cells and CD4 helper CD4 cells. So interleukin 2 pro-inflammatory has nothing to do here. Interleukin 12, again you know that interleukin 12 is actually responsible for activating the acquired arm of the immune system. So that is also pro-inflammatory. It has nothing to do with the anti-inflammation. So not going to be useful here. Interleukin 4, that was the option number C in the question. Interleukin 4 and 5 are actually released by T helper 2 cells, and these act on the B cells and activates them and causes them to do class switching and release more antibodies and so on, and we'll talk about those when we go to the B cells. And finally is interleukin 10, and bingo, interleukin 10 is the answer. So I hope you enjoyed the question. Nice little story, but a very important thing. Tumor growth factor beta and IL-10 are anti-inflammatory proteins. Do not forget them. Thank you very much, and we will continue.